Welcome everyone to the FASEB webinar, How Co-Funding Mechanism Supports Early Career Investigators. I'm Naomi Harlem-Bacchus. I am joined today by Dr. Eliseo Castillo and Dr. Nabila Rias, who is also at FASEB, and we are delighted that you are here today for the second webinar in our series on the IDEA and EPSCoR programs. So if you haven't seen the first webinar in the series, which happened a couple months ago, you can go to the FASEB website and go to the events page, past events, and you can find the recording and slide share where we talk about the COBRI mechanism that took place in Mississippi and all of the great work that they're doing. So before we get started, what we're going to do is I will share a little bit about the IDEA and EPSCoR programs and a little bit about FASEB, and just to get everyone oriented before we present we give the floor over to Dr. Castillo, and he can give his presentation on how the IDEA program has benefited him and his career. So again, thanks so much for tuning in. Let's go ahead and get started. If you're not familiar with FASEB, FASEB is the Federation of American Societies for Experimental Biology, and we are comprised of 22 member societies that you can see listed here. And this spans a broad range of different scientific disciplines, which makes it really great working at FASEB because you get a broad understanding of so many different areas of science. And we represent over 110,000 scientists, engineers, and biomedical researchers. Moving right along, so for today, our agenda is we'll give a brief overview of the NIH IDEA and NSF EPSCoR programs, which are comparable programs. And today we will specifically focus on the co-funding mechanism. And of course, our speaker, Dr. Eliseo Castillo from the University of New Mexico, will share a specific insight into how the IDEA program has really benefited his career. We will have some time for Q&A at the end of the meeting, so please feel free to submit your questions. We will get to as many as possible. And on that note, just some housekeeping. We are in a webinar format, so that means the chat and the raise hand functions are disabled just for logistical purposes to make everything easier. But please submit your questions in the Q&A box that you see highlighted here with the red arrow. We will get to as many questions as possible at the end. Please note that we are recording this webinar, so if you're not able to tune in for the whole thing, or if you want to watch it again because it was so riveting, you can definitely find the webinar on the FASEB website in the next couple of weeks for distribution or for future purposes. So we will also be attaching Dr. Castillo's presentation as a PDF where you can find all of that information from his talk today. So without further ado, let's go ahead and get into just a brief overview of what the NIH IDEA and NSF EPSCoR programs are, which is what this FASEB webinar series is all about, is really highlighting the great successes that have come from these programs. So if you're not familiar with the IDEA program, the IDEA program stands for Institutional Development Award, and it was actually established by Congress back in 1993. And the purpose of the IDEA program is to broaden the geographic distribution of NIH funding, particularly for states that have historically received lower levels of NIH funding. So all of the states that you see in blue are considered IDEA states, which makes it roughly 23 states in Puerto Rico qualify for any of the benefits as part of the IDEA program. The IDEA program is administered by one specific institute at the NIH, in particular, the National Institute of General Medical Science. They always issue the awards, and those program officers can really be helpful if you have any questions about the IDEA program. There are 5 main components to the program, and you see them all listed here. The first one is the COBRI mechanism, which stands for the Centers of Biomedical Research Excellence. And this is really geared to supporting the development of multidisciplinary research centers and really fostering that collaboration across different scientific disciplines. The second one is the IDEA Networks of Biomedical Research Excellence, or INBRE, and this is geared for supporting more faculty and postdocs and graduates, so really building up the workforce. The third one, which is what we will focus on today, is the co-funding mechanism. And this is support for research project grant applications from investigators whose proposals received meritorious, very high scores in peer review, but they fell just short of that institute or center's payline. And Dr. Castillo will share a lot more about how that process plays out in real time and during study sections. The fourth is the clinical and translational research programs, and this is helping states develop their competitive clinical and translational research programs by supporting the infrastructure and human resources development. And then finally, the IDEA Regional Entrepreneurship Development, or IRED. And this helps build up the small business opportunities in these states and develop those educational and promotional products. So those are the 5 different components. There are a wide range of different funding opportunities through these different mechanisms that have really enabled the IDEA program to benefit these states in particular. Similarly, oh, and here I have my animation that I completely forgot about, but you see that today we are going to talk about co-funding, which is why I have that underlined. The other program that is very, very similar, just is administered by a different federal agency. The National Science Foundation has the Established Program to Stimulate Competitive Research, or EPSCoR program. This was also established by Congress actually way before the IDEA program in 1979, but a very similar purpose: to bolster research competitiveness of designated jurisdictions. So it's not really called EPSCoR states, they're called jurisdictions, by once again strengthening the STEM capacity and enabling improvements in their research and overall infrastructure. It's administered by the NSF Office of Integrated Activities. And from this map here, again, you see in blue all of the different EPSCoR jurisdictions. There are a total of 28 that are eligible for EPSCoR benefits and funding, as well as Puerto Rico, Guam, and the Virgin Islands. So there are 3 investment strategies that are pertinent to the EPSCoR programs. The first one is called the Research Infrastructure Improvement Awards, or the R2 Awards, or RII Awards, either or. This helps build the infrastructure and workforce development for each of these EPSCoR jurisdictions, again, emphasizing collaborations and team science. The second one, and you'll see a theme here with the IDEA program, is co-funding of disciplinary and multidisciplinary research. And this is where NSF co-funds proposals that are submitted to other NSF programs that are recommended for award, but they fall just short of those budget means. So other directorates chip in to help get that funding completed. And then the third one, which I find is really interesting with the NSF EPSCoR program, is they fund workshops and outreach. So they really encourage different EPSCoR jurisdictions to fund collaborative activities in your jurisdiction to explore opportunities in emerging areas of science. Oftentimes, these emerging areas are really relevant to the state that you are in and the specific problems that that state is facing. So that is a little bit about the IDEA and EPSCoR programs. Without further ado, the main event today is our speaker, Dr. Eliseo Castillo from the University of New Mexico. He's an associate professor and the director of gastroenterology research at that university in the Division of Gastroenterology and Hepatology. So we are so excited to have you today, Dr. Castillo. And without further ado, please take it away.
Thank you, Dr. Hollenbachus and Dr. Ruiz and the FASEB. I'd like to thank all of y'all for allowing me to give this talk and to present my R01 application and how the IDEA was involved in this funding source. Actually, after that brief presentation of about the IDEA states, I realized I have benefited from 4 of these IDEA programs. The COBRE, we have several COBREs at the University of New Mexico. I have received pilot funds from the COBRE. Actually, they assisted in getting this R01. I was a CTSC KO2 scholar. And as an undergrad, I remember doing research in an MBRE program as well. So I think this has been very beneficial, and I'm looking back now and seeing how much I've benefited from this program. So today's talk is discussing my R01, defining the harmful effects of microplastics in GI health. So just real quick, starting my own lab, I started my lab in 2017 as a CTSC KL2 scholar. I highlight this in red because I think if there's any postdocs looking for faculty positions, you should actually look at to see what universities have CTSAs and CTSC KL2 scholars. I know UNM HSC has one, and we actually have KL2 scholar spots that are open to external and internal applicants. But I got my start, and during this start, it was to actually examine autophagy and macrophages in inflammatory bowel disease. I started off with one technician. During this time, I submitted an RO1, and then during that RO1, I got a comments back. Basically, the reviewer said, autophagy and IBD has been done. This is not novel. So how did I move forward? Around that same time in 2018, there was an article that came out that this was discussed in my division, the division of GI, about how microplastics were found in human stools for the first time. This was a gastroenterologist in Europe, and basically he collected stool samples from various countries from where he was at in Europe, looked at the amount of microplastics found. I applied for a pilot project through one of our COBRE systems here at UNM to actually assess what is going on if mice were to receive microplastics, and does this impact GI health? So with that pilot study, this led to me submitting my first RO1 on microplastics in GI health, and this was in October 2019. I received my score back, and this was about, let me see, in March of 2020, and I received a 38th percentile. I was pretty excited because I actually got discussed. However, there were some major issues that reviewers brought up. It was stated that I had no papers on microplastics, which was actually very true because I was pivoting with my research from macrophages and inflammation to now microplastics. This grant also went to NIEHS, which is the National Institute of Environmental Health and Safety. And obviously they're big on toxicology, and it was suggested that I needed a toxicologist on the grant as well. It was also suggested I needed a model to examine different doses. So what I did do is actually, after I received my score, looking at NIH Reporter, I saw that there was no grants on microplastics, and I thought this was a new untapped area of research. It was getting some news. And so I reached out to the program officer to discuss getting an R56 application. This was a simple email that I sent to discuss with the program officer to discuss that if I can get an R56. It was nominated by the NIH staff, and I received an R56 on September 2020. So you can see my initial application went in October 2019. So this is almost a full year around. So during this time, we did publish a short communication article to sort of respond to the reviewer comments that I do now have a paper on microplastics. I included a toxicologist. This was also just to show that the toxicologist who would now go on my resubmission, that we are working together. And I met some new collaborators to sort of utilize a new model to actually examine the various doses. This was actually Dr. Sung Sai from UCLA. It was a quick turnaround for my R56. I got it, as I mentioned, in September 2020. In March 2021, I resubmitted my A1 application. The payline was still at 11th percentile. In this application, I responded to all the reviewer comments. At least I tried to. I added a new aim using zebrafish to examine the dosing as the reviewers requested. I added a toxicologist. I also highlighted that I had published on microplastics. In July 2021, my summary statement, I received it. I got a 12th percentile impact score of 28. At this time, I was actually, you can see the pay line was the 11th percentile. I was 1 point out of the pay line. So what was my next step? I had learned a lesson previously when I first started in 2017, or I apologize, 2016. I submitted a K award, And the K award, my impact score was a 27, and the payline was, or the impact score payline was a 26. I was unaware that I needed to talk to someone higher up in my academic institution to see if they can sort of push this over the payline and get us funded. But learning my lesson, I reached out to Dr. Larson, who at the time was the executive vice chancellor and the vice chancellor of research at UNM-HSC, to discuss my options. I did email the program officer and mentioned that the University of New Mexico Health Science Center was part of the NIH IDeA states. At that time, Dr. Larson was in charge of both the CTSA and the CTSE Scale 2 Scholar Program. So he was in the know that I actually was submitting an R01. He was in the know that I got a 12th percentile and that the pay line was 11th percentile. I bring this up because there's several faculty here who have been in a similar situation as I was, where they were right outside the pay line. However, they did not know who to talk to. So me being in the CTSC was sort of this perfect timing. Me knowing Dr. Larson, I could easily walk down the hallway, discuss this with him. I know that's not an option for everybody, but I do think if you're a junior faculty just starting out, it's very important to reach out to find out, hey, if I'm ever in this situation, who is the person to talk to? We did find out there is a deadline. It's not advertised. So find these key information out when you're submitting your grant and you get your score back. It took a full year since my initial March 2021 to finally receive, over a full year to receive an NOA. And this started in September 2022. I am now in my third year of this funding. How much did my A1 submission change? It actually did change quite a bit. And this was due to the response of the reviewers. We brought in a zebrafish model, and we also brought in a human intestinal organoid model. One of the reviewer comments can we get this related more to humans? And so using this human and intestinal organoid model was very important. Some of the things that I did establish is a collaborator who studies zebrafish. I also have a close collaborator that's in my division who is an expert in human intestinal organoid models, and I have brought her in on the team as well. So I would say responding to the reviewer comments, building a nice team was very important, and I think actually helped me get pushed over the line to get paid, as well as being part of the IDeA state. Currently, right now, well, I would say for the last few years, I have been the only R01 who has been studying microplastics on any type of human health. I have utilized the diversity supplement to pay for one PhD student. So if you have an R01, this is something you should look into, because these diversity supplements actually help with individuals who are from historically underrepresented populations. As soon as I published my paper, as soon as I got my grant, there's been an explosion of microplastic research, especially at this campus at the University of New Mexico. A colleague of mine in the Department of Biochemistry just received his NOA this past month, and it is now up on NIH Reporter. He now has an R01 as of August 14th. There are several R21s that are also examining microplastics, but this is a new field. I will say, as a young investigator, I had to pivot, seeing that my macrophage IBD story was immediately tore apart. And it is a hard field to step into, but I think this pivot, utilizing these pilots, these internal pilots, actually helped me out so much. These are the current publications from my current grant. We are currently at 5 publications. This one's currently under review at Nature Medicine. We are excited for our research. I will be happy to take any questions on this application process, but I do sort of want to discuss what I am doing with this grant. As I mentioned, in 2018, this first article came out talking about discussing how they found microplastics in the human stool. Getting into the literature, it has been reported and is constantly reported that the average human consumes about 5 grams of microplastics in a week. That is equivalent to a credit card. Over the last, I would say, 5 to 6 years, there's a plethora of papers that have come out, and the major route of exposure is actually through the gut after the consumption of food and water. Drinking and eating have been classified as the most common exposure type by WHO. So what I'm going to show you right now is this sort of our recent findings published in the Environmental Health Perspectives, and also show you some unpublished work and future plans. So we just published this paper back in April. It was in Environmental Health Perspectives. And the major findings of these paper was after mice were exposed to polystyrene microplastics, we can find microplastics in the brain, liver, and kidney after oral exposure. When we looked at the brain and liver as well as the colon, we found this plastic exposure led to metabolic changes, and we tested this by metabolomics. And we also saw there was a differential response dependent on the concentration and the type of microplastics the mice were exposed to. This was a large experiment. There were 5 groups where mice were exposed to the 2 mg per week, which is basically the credit card worth of microplastics that humans are thought to be exposed to, or we doubled it. The reason why we doubled it is because it's thought that the credit card's worth is actually an underestimation of what we're actually being exposed to. And this was a single polystyrene, 5-micron polystyrene microplastic microsphere. We also exposed mice to a mixture of microplastics that had different sizes. These are the mixtures. It has 1 to 4 micron polyethylene microplastics, 5 micron polystyrene, or 5 micron PLGA, which is actually a biodegradable microplastic or plastic. And we also increased this dose. So after a month of exposure, we wanted to collect all the tissue and determine a way to detect if there's microplastics present. This is a brand new field, and there was no ways to actually detect, or there was quantifiable ways to detect microplastics. So we devised a methodology to actually do this. This was published in Toxicological Sciences, where we could isolate a piece of the tissue and have it undergo saponification. This will lead to the isolation of these microplastics, and then through pyrolysis GC mass spec, we would see if there's microplastics in the tissue. This is after the 4 weeks. This is just looking at 2 mgs per week of the polystyrene, or the 4 mgs. When we examined the brain, we were able to detect small pieces of microplastics. Same thing for the liver, and the same thing for the kidney, and it could also be found in the serum. I know you're saying these are probably just specs. We wanted to ensure, so we actually did Raman spectroscopy, and what we did is we took the standard film, the 5 micron beads, and we looked at the fingerprint, and then we took the brain isolate to actually see, are these polystyrene microspheres? We also treated them as we would treat the beads with this KOH, and we found that it does actually modify it, but not by much. So we strongly believe that this are microplastics found in these different tissues. One of the reviewers did come back and say, I don't believe that those are microplastics in the brain. And that's an astonishing finding. It's an astonishing claim to make. But we did do metabolomics, and one of the things, and first I'm going to show you the colon metabolomics, we did see different metabolites present after different exposures. These are all compared to the control mice. There are some overlaps, but when we're looking at this, these were different pathways that were present or that were upregulated in the colon of these mice after exposure. And we did the very same thing for the brain. And something that we highlighted to the reviewer was, yes, you're absolutely right. We did our mass spec. We did everything control as suggested. But one of the things we wanted to highlight, just the exposure alone, regardless if we didn't look for microplastics in the brain, is that we are seeing metabolic changes in the brain after the exposure. So one of the questions that has come up, and I will bring this up right now, is where is it at in the brain? Is it in the tissue or actually in the brain vasculature? Is the plastic causing metabolic changes in the brain, or is this due to possibly changes in the microbiota? Do microplastics cause inflammatory bowel disease or neurological diseases? There's so many questions that I get when I discuss our research. The list goes on and on and on. But with this study, we were able to detect polystyrene microspheres in the brain, liver, and kidney after oral exposure for 4 weeks. This did cause metabolic differences in the colon, liver, and brain to unexposed mice. And the response is actually dependent on the concentration and the type of microsphere. As you can imagine, we're probably getting exposed to various types of microplastics, different sizes and at different doses, depending on where we live, on our diets, etc. So there's a lot to be explored in this field. So one of the questions I get is it actually breaking down the barrier, and is it going to different places? How is it passing the GI tract? Once it passes the GI tract, what happens? So to sort of understand this in the different sizes of microplastics, we have our collaborators, a sub-awardee from UCLA, Dr. Sung Shai and his lab, and postdoc Suki Park, and where we're microgavaging embryonic zebrafish. And what we do is we actually microgavage the microplastics in the presence of this di-ritzy-dextran at 10 kilodaltons. And we wanted to understand the exposure. So here are control mice with ritzy-dextran. And we see very, very little ritzy-dextran that has passed into the abdomen. When we give the ritzy-dextran with fresh, pristine polystyrene microplastics, we can see a hint of it that is actually being exposed in the abdomen. When we inject the ritzy-dextran with EDTA, which we know will actually break down the gut vascular barrier, we see a lot of the ritzy-dextran accumulating here. And this is basically the venous plexus. We have photo-age microplastics because this has also been another, I guess, a major point of interest for microplastics research. We're using pristine, but probably what is out there are damaged UV-age microplastics. So we're trying to get more environmentally relevant. So we photo-age these polystyrene microplastics for 4 weeks. And what we find is that the photo-age polystyrene microplastics does lead to a disruption of the gut vascular barrier. And we see ritzy-dextran accumulating here, similar to our positive control of EDTA. Where the fresh polystyrene microplastics has somewhat of a disruption, but not as much as the photo age. We also wanted to see if ritzy-dextran are going to other places after this exposure. And this is looking at the heart as well as the brain. And we can see ritzy-dextran going into the heart and brain, the photo age exposed mice. We are trying to understand the mechanism of how the photo-age polystyrene break down the barrier and leads to this disruption. The next step of ours is also to understand diet microplastics and basically what is going on in the colon. A major criticism that I've received, and I understand this criticism, I sort of agree with it, is we have been orally gastric gavaging mice with a bolus of microplastic. And this is not how humans are normally exposed. So what we wanted to do is actually incorporate microplastics into the chow of into different chows of mice, and then expose them for a longer period of time. As you can imagine, oral gastric gavage is very stressful on the mice. So we have set up an experiment where we have a normal chow plus microplastics, high-fat diet, the high-fat diet plus microplastics, as well as a high-fiber diet with different sizes of microplastics. We have exposed mice for 12 weeks. This is an experiment that has completed, and we are now in our final phase of this huge project. And we're looking what's going on with the mesenteric fat as well as what is going on in the colonic microenvironment. So we look forward to putting this together in the future, and hopefully you'll see something out from our lab for the zebrafish as well as these mice. Why is it important to look at the high-fat diet or the high-fiber diet? Also, what's going on in the barrier? The reason why is because we study organoids, and we actually have found that when you take organoids, and when I'll explain what an organoid is in a minute, when you take organoids from an individual who is healthy, they will actually have a healthy intestinal organoid cell line. But when you take organoid, or you grow organoids from someone who has inflammatory bowel disease, it actually represents that disease. It carries the phenotype in vitro. So an organoid is, and the way we do it is we actually get tissue biopsies from someone undergoing a colonoscopy. We isolate the crypts, specifically the intestinal stem cells, and then we grow them in matrigel, and we can keep these organoids in a stem state manner, and so they will be intestinal stem cell organoids. Or we can remove certain factors to allow them to differentiate to become different populations of a mature epithelium cell. And this is what we see here, is basically an intestinal stem cell can become different types of epithelium, your basic epithelial cell, a goblet cell that makes mucus, enteroendocrine cells that makes hormones, and Panna cells for the small intestine. So we have other research projects showing that individuals who are on a high-fat diet, that there is a skewing of this stem cell differentiation when compared to mice on a controlled diet or lean mice. So what we want to do is sort of grow these lean-derived or high-fat diet derived colonic organoids, make a monolayer, because we know we will see differences in different populations of cells, and see how microplastics, this organoid barrier, intestinal barrier, will respond to microplastics. Will a high-fat derived organoid lead to an increase in microplastic passing through the gut barrier, or will the lean derived one actually stop some of the microplastics from going through? These are all future directions for our research, and we're excited to keep doing this research in this field of microplastics. So one thing I want to plug in before I finish is I am part of the University of New Mexico Comprehensive Cancer Center program to enhance diversity. Our mission is to serve the unique, diverse populations of the state of New Mexico, and to ensure that the center's faculty, staff, trainees, and leadership is representative of the population it serves. And one of the major aims that I have for this program to enhance diversity is to recruit early career scientists and clinicians. And you know, you can reach out to me if you fall into these categories, but we will begin starting a Rising Star lectureship, where we would want to invite K99s or other K awardees to present their work at the University of New Mexico's Comprehensive Cancer Center. This would allow you to give a talk, tour our facilities, meet our faculty, and possibly so we can recruit you. We are actually interested in individuals who have a major focus on cancer research. As I brought up earlier, we have this CTSA program here. The CTSC is now going to be called the K-12 program, and this is another potential way to recruit young investigators. And we are actually interested in individuals who are have a focus on cancer research. The CTSC K-12 program, the past program was a 5-year program where you bring in individuals who have a research plan, and you are taken care of by the department. 75% is paid by the department, 25% is paid by the CTSC, and you have 5 years to sort of work on an R01. You get mentoring. There is a huge committee that also helps in your mentorship. All of this is to help you to be successful and to get an R01 award. So if you do have an interest, or if you fit into one of these categories, please reach out to me. My email is on the top slide. It could be, or I can provide it later. But this is something to, if you are on the market or going to be on the market soon, please look out for this because we are interested in recruiting individuals to the University of New Mexico. And with that, I want to acknowledge all the individuals in my lab. We have Aaron Romero and Diego Ruiz, who are working on microplastics. Some former members include Seth Merkley, he was a technician, and a medical student, Jewel Meyer-Hagen. Both of these individuals were the one who initially started the pilot project and got everything going. So I owe a lot of thanks to them. Also, the various collaborators, Dr. Julianne, she is the expert in organoids. Sung Sai and his postdoc, Dr. Suki Park at UCLA, working on the zebrafish. A major collaborator here as well has been Dr. Matt Campen and his lab, postdocs Marcus Garcia, Samira Fitek. Both of these individuals have helped with the animal studies. There are also a major group who has been doing this pyrolysis mass spec to identify microplastics. And of course, I would like to thank my funding sources. NIGMS, the IDeA, they're the ones who got this started for me. I also have a P42 and a P20 award, and the P20 award is a COBRA. I also like to thank FASEB and Dr. Harlan Bacchus. And with that, I will be happy to take any questions.
Fabulous. That was completely fascinating. And I have questions about your research, also questions about how you got the funding. So thank you so much for taking the time to walk us through, especially your career path. I think that was really helpful for our audience. So lots of questions to get through. First one, is there a clear distinction regarding how far a principal investigator can be from the institutional payline in order to be considered for COBRA co-funding?
That is interesting. I have seen individuals, actually the recent individual who received an R01 at the University of New Mexico, I think he was at a 14th percentile. And the pay line was still at 11th. So you can see there was a 3-point difference. I think that's at the program officer's discretion. Yeah. I will say another thing is though too, for young investigators who are putting into NIEHS, their program is different for early stage investigators. There is a specific FOA that they have to apply to be considered ESI, where other institutes such as NIDDK do take in consideration your ESI status. I was a new investigator, and in my resubmission, it was a month after my ESI status had finished, so I didn't actually have that ESI status.
Gotcha. I was going to say, I think it's dependent largely on the IC as well.
Right.
So everyone is different. That's really helpful though. Thank you. Because it can be 1 point difference, 3, 4. So
Right.
it is up to the PO. What were the challenges in moving from the R01 to the R56?
So I would say, so for my initial submission, I had to focus my R56 on something the reviewers really wanted as preliminary data. So that was, you have yet to show microplastics cause a change in metabolism. And that's why we focus on 2 things. We focused on understanding, was there a change in an immune cell such as macrophages, and it did alter the metabolism, and did it alter the metabolism of epithelial cells? And we showed both of those in the R56. And that generated preliminary data to go into my A1 submission. So I think it's sort of picking, what can I utilize this R56, these funds from the R56 to best help my A1 submission. And if it's something the reviewers are asking for, you need to provide this type of data, there needs to be an animal study or whatever the case may be, that's my recommendation, is to go after what the reviewers really want.
Yeah, that makes sense. And that actually goes in nicely to one of the questions I had is, in one of your slides, I liked how you phrased it, is you sent a brave email to your PO. So what is your advice to someone that maybe is too afraid to send that brave email to the PO about their applications and potential next steps.
Yeah, so I would say one of the things I was told was wait till my summary statement come out. Wait till the summary statement comes out, and as soon as it comes out, look at the reviews, and it's pretty positive. You feel like the overall reviewers were pretty positive. It's just somehow the scores got lost in in the reviewing process. I also think no one's going to speak up for you more than you can for yourself. So I do think you just have to it's a simple email. All they can do is say no. And so my email to them was, hey, I got my score. The reviewers were very positive. I want to ask about the potential of getting an R56 to address some of these reviewer comments.
Yeah, I love that. That's great advice, especially for folks that are starting out in their career and don't know where to go and what's okay and what's not. How can I find out who to talk to at my university about nominating my grant for potential co-funding?
Yeah, so for me, this was sort of obviously I was at the right place at the right time, but Dr. Larson is actually retired now, but I know there was faculty here who really never had a chance to meet Dr. Larson. So I had the opportunity because I was a CTSE K02 scholar, and my office was literally down the hallway from his. So I would see him on a daily basis. But these are things when I guess I would go to someone who is very well funded in your department, whether that's the chair or other faculty members, to say, hey, I'm getting ready to submit my R01. If I get a good score, let's just be positive. If I get a great score and but it's out of the pay line, what is my next step? I have heard there are situations where the university, since it's an IDEA state, can push it forward, try to get it funding through NIGMS, but it's just basically asking the questions. As I mentioned earlier, I did apply for a K22. 27 was my score. The impact score that the pay line, I guess, was a 26. I heard from the program officer, but I didn't know who to reach out to. I didn't even know to discuss this with the chair of my department at the time. But yeah, I would just say I would start asking individuals who are funded in your department or your institution, talk to your chair. And then also if there's a sponsored projects or grant submissions, who else you can reach out to them to say, who is an individual I can talk to about IDEA State and NIGMS funding.
Great. Yeah, I was going to say, I think sponsored projects offices can be really helpful for issues like this too. Thank you. That was great. Is co-funding applicable to both new investigators and early stage investigators, or only to the latter, early stage?
I actually thought it was for both.
My understanding is it's for both. It's for applications that are just shy of the pay line, and if you're in an IDEA state, then you are eligible to apply for co-funding. So yes, not just early stage. The next one, when moving forward with the R01 rejection, what are the options other than R56?
That's a hard one. So I had other colleagues here at my institution suggest I submit an R21. But when I talked to the program officer said, I really like this grant. I think you should go back in as an A1 submission. So, I mean, there's R21s that you can actually pull this apart. I also think it sort of maybe there's possibly NSF funding that you can go to, but the NS funding, I think that's dependent on what type of research you do, because I know they take only certain types of research. So there's NSF options. You can also always apply to different types of foundations. If this would be applicable for me, you know, I studied the gut, I could have went to AGA. So also looking at foundations as well.
Yeah.
But I think talking to the program officers, seeing if you got a good score, it's not in the pay line, talk to the program officer, is this worth putting in an A1 application?
Right. I think having that conversation is essential. I think
Yes.
that's great. Does the IDEA co-funding mechanism apply to all grants or just certain grant mechanisms?
If I'm right, I thought it only applied for RO1s.
It's it's
Actually,
actually, yeah, go ahead.
R35s as well.
Which ones?
I think in R35,
Yes, the R35, which is the mirror mechanism, as well as R15s.
R15.
So R01s, R15s, and R35s.
Okay.
Oh, yes. Do you have, this is a good question. Do you have any suggestions on how IDEA states can improve grant applications? Are there certain things to emphasize versus non-IDEA states?
I am I am on a study section. I am a standing member for a study section. And I can say these are things that are not discussed.
Hmm.
Like, are you an applicant from an IDEA state? So I don't know if there's a mechanism to say, hey, um, my suggestion would be, is there a study section specifically for IDEA states? I don't know how that would work because you are going against every other, every other applicant from everyone else from across the states. But yeah, I just, the other thing I would suggest is, if I'm right, I think only institutions could put in an application once per year. So maybe increasing the amount of applications they can put forward to the council for
Yeah,
NIGMS.
yeah. Yeah, there is that limit. I think the limit varies depending on the university actually, but it's usually very small. It's like 1 to 2 applications per year. I know, for example, UNM has a limit of 1. So yeah, increasing that limit would be helpful. What happens to my co-funded grant if I get another position and then move to a non-IDEA state?
I do not know the answer for that. I mean, I know the NIGMS covered the first 2 years of my grant, and years 3 through 5 are now covered by the sponsors NIEHS.
Gotcha.
So I'm completely unaware of what would happen in that scenario.
I believe my understanding of the IDEA program is that if the part that NIGMS is funding and you are now in a non-IDEA state, those funds would be terminated because it's predicated on you being in an IDEA state.
Okay.
So that's my understanding, but we can certainly double check. Is it possible to remain a COBRE PI once you receive an R01?
Actually, it is not. So both COBRE, if you're an MPI on a COBRE or you're a CTSC KL2 scholar, the goal for both of those, at least at our institution, is to get you an R01. Once you become you get R01 funding or R01 like funding, you actually graduate from both the CTSC or KL2 program as well as a COBRE program. You're also not eligible to be a COBRE MPI or a CTSC KL2 scholar if you have R01 or R01 like funding.
Gotcha. Okay. That's very helpful. Okay, I believe this is the last question, but we still have time left. So if folks still have questions, please feel free. You mentioned applying for pilot projects at your university's COBRE program at the very beginning. How can we find out what projects are going on at a university?
Yeah, so fortunately for us, we have I think 2 or 3 COBREs. I know for sure 2. I don't know if the third one has ended. But they do send out regular emails to say our centers, or we have also program awards that send out emails to us that essentially state, we have these pilot projects for 1 year for X amount of dollars. The CTSA here as well at the University of New Mexico CTSC program, they also have a pilot program. But I mean, I think this is actually possibly talking to the Vice Chancellor of Research at your institution to see, are there pilot programs at the institution? Who's eligible to write one? Because sometimes you actually have to be a faculty member to be eligible to write where some of the programs at UNM have actually allowed postdocs to apply for some of these
Okay,
awards as well.
yeah, that's really helpful. And then someone messaged us and shared, and I'm going to put it in the chat for folks that want to learn more about the co-funding program. There's a great website through NIGMS about eligibility and how to contact your program officer. So please feel free to check out that, because that's a great resource if you're thinking about, applying for a grant and you fell into a similar situation as Dr. Castillo, and it was just shy of that pay line. So you definitely have options. So that was helpful. Thank you. I believe those are all the questions. Folks still have time. We have 10 minutes remaining, but I do want to be respectful to give everyone else back time if you want, because time is always short nowadays. But Dr. Castillo, is there any other last minute thoughts or advice that you would like to share before we wrap things up?
No, I thank you for the questions. This is I'm learning as well. And as I move up and the academic ladder to be as an associate professor, there's always new faculty coming in. But, I do think taking advantage of every situation that you can, whether it's applying for the IDeA state, looking for COBRAs, just trying to utilize every resource to be successful. It's very hard to get grants these days, so utilize these resources.
And as you said earlier, no one can be a better advocate for you than you, and speaking up and really leveraging the resources that you have and talking to your program officer. I think those are essential. So FASEB is trying to build up more awareness about the IDEA and EPSCoR program. So please feel free to reach out if you have additional questions, and you have great advocates like Dr. Castillo, who has been, really very wonderfully come today to share about his career path and ways that others can do the same. So I really appreciate everyone's attention today, all of these great questions and your time. So again, this webinar is being recorded. It will be posted on the website in the next 1 to 2 weeks or so, along with Dr. Castillo's presentation. So his email address is included at the very beginning, as you saw. So you can feel free to contact him or myself at any time. So I believe we can actually, I think one more question came in. Is there any contact information available at NIGMS for co-funding? Yes, you can. I will put that in the chat as well. There is a dedicated contact person, Dr. I will not be able to pronounce his name correctly, Dr. Yang Zhu over at NIGMS. He is directly in charge of the NIH IDeA co-funding initiative. So please feel free to contact this individual that I just included. That is your go-to person if you have specific questions about co-funding. So I believe, and that's a great way to end it, always with email address and way to find more information. So thank you again, everyone. Thank you again, Dr. Castillo. And I hope everyone has a great rest of the day.
Thank you so much. Yeah, have a good one.
Bye.